and his clinical position as a head and neck surgeon there, and then has been really successful in getting funding, not only in a research program about the referral to and fast track referral to specialist services, but also, and this is what he's going to talk about, I think, also the management of recurrent head and neck cancer, which apart from drug treatment and chemotherapy, I think is by and large a bit of a Cinderella subject. So we look forward to Vin next week. Now, just while everybody's been joining, what I will do is just introduce Tim. So Tim Budden is a proper doctor, not like those of us who are just merely dentists or doctors. Tim joined us very recently, and Tim joined us in molecular clinical cancer medicine. And we have, I think, successfully inveigled him in the Head and Neck Centre and the Mersey Head and Neck Oncology Research Group. But in fact, his background has been in cancer research in a diverse area. Tim's background academically was in Australia, the University of Newcastle in Australia, not Newcastle in the northeast of England. And at that point, logically enough, he was studying malignant melanoma, DNA damage, and in part as well, the role of DNA damaged fibroblasts in terms of treatment resistance, I think. He had postdoctoral positions in New South Wales and Sydney. He's also been involved in work in other skin cancers, ovarian cancer, and lung cancer, a number of very high profile publications. He moved about 6 years ago, obviously looking for better weather, to the northwest of England. He's worked for about 5 years in Manchester at the CRUK Manchester Institute. I think in that point, mainly again on skin cancer, melanoma, and I'm still working on ultraviolet sunlight damage and fibroblast behavior. And then about a year ago or so, I think moved to Liverpool University for a lecturer's position. So I'm going to stop sharing that. I presume while I've been talking, and I can see people have joined. And so, Tim, it's an absolute delight to hear you tonight. It's always brilliant to have new scientists join our group, and over to you.
Thanks. Let's share my screen. Can everyone see that all right? Yeah, okay, good.
It looks great, Tim.
Yeah.
All good.
Thanks, Richard, for the introduction, and thanks for inviting me to give a chance as a new scientist at Liverpool to present some of my work and also what I guess plan to be working on as I set up my group. So yeah, thanks. As I mentioned, my interest has been mostly focused on fibroblasts in my research, and so tonight I'm going to present a bit of background on what I hope to do as I move forward into looking how we can target these fibroblasts in head and neck cancer prevention. So yeah, a bit more of a scientific background than clinical. So my work is kind of about how we can understand fibroblast biology to sort of design new cancer prevention strategies. And this has kind of informed a lot on my research of how fibroblasts impacts outcomes in cancer. And as Richard mentioned, most of my work in the past has been on skin cancer. So I'll go a bit into this to sort of show the ideas I've learned working in skin cancer. But as I've been moving forward with my career, I've sort of been applying this more broadly to looking at how the biology of fibroblasts sort of is tissue specific and how we can target these fibroblasts and their specificities across different epithelial cancers. So I'll try and focus mainly on oral cancer, but I will also touch a bit on some lung research that I do as well. So just to give a bit of a background for anyone who may be unfamiliar, epithelial cancers are extremely common solid cancers. Of course, they derive from the epithelial cells that actually are present across multiple organs. So you can have epithelial cancers in the skin, oral mucosa, respiratory and digestive tracts. I mostly focus on a type called squamous cell carcinoma, which I'll refer often to as SCCs. So these are just epithelial cancers that are derived from squamous cells, which again occur mainly in the skin, oral and respiratory tracts. So these are usually classified by anatomic location. So they're referred to as things as cutaneous SCC for skin cancer, lung SCC, and head and neck SCC. But they actually have quite a lot of overlapping features and some common histopathologies. So epithelial cancers, a lot of them can be driven by chronic exposure to environmental carcinogens. So this is things like tobacco, cigarette smoke in terms of lung cancer, alcohol and smoking in head and neck, or ultraviolet radiation from the sun in skin cancer. So there's a lot of work in these fields, in these cancers, in prevention in terms of things like anti-smoking and sun safety campaigns that I'm sure we're all familiar with, to reduce the risk of people developing these cancers just through reducing exposure. But these preventions can also be screening programs, so looking at patients or people who are at high risks of developing cancer and monitoring them. And these could be stratifying risks of development, such as through behavioral risk factors like smoking or genetic risk factors. But the side of prevention that I'm sort of more interested in is understanding tumor biology to see if we can identify biomarkers, so things that we can look at in patient samples that might be able to predict the risk of them developing cancer or progressing into more aggressive cancer. And then by understanding the biology of this, can we find new targets of therapy to prevent or development or prevent progression? So as I mentioned, epithelial cells across all different organs, but they have a relatively similar structure between different tissues. So for example, here we've got a section under the microscope of skin and of the oral mucosa, so the lining of the mouth. And you can see up top here in the purple is the epithelial cells. So you can see there's a bit of variation between tissue. And these are the cells that form the cancer. But what I'm interested in is what's going on in the stromal layer. So this is often referred to colloquially as the connective tissue. And it is the section here in pink. And this is where we see the cells such as the fibroblasts. It is generally made of extracellular matrix proteins and is also the location of blood and lymph vessels. So fibroblasts are a pretty common cell type. They're found in almost every tissue that has connective tissue. And they're actually quite important for maintaining tissue health. They do a lot of things, but to simplify, the main things they're known for are being heavily involved in wound healing. So these will contract and tighten the wound and replace the damaged proteins. They're very important in producing and maintaining the extracellular matrix. So these are the proteins that sort of provide structure and shape to our tissues. There's things include, everyone's probably heard of collagen. It's one of the major proteins, but also things like fibrin and elastin. And this is important because it forms a barrier in the tissues, but also is involved with the cell attachment, movement, growth, differentiation, and repair. It's very important. It could be a whole talk, but I won't go into that. And they also have a role in immune regulation and immune cell recruitment into the tissues, again, in things like wound healing. But they also play a big role in cancer. So a lot of research is focused on what we call cancer-associated fibroblasts, or CAFs. So this is where cancer cells kind of co-opt the fibroblasts into the tissue and sort of convert them into an evil cell where they are pro-tumorigenic. So they can do things like remodel the extracellular matrix to promote invasion and metastasis, the spread of the cancer. They can prevent immune cells or drugs entering into the tissues, which inhibits therapy, and they can be quite immunosuppressive. So down here we can see in a normal tissue here, which I believe is pancreas, the pink here again is the stroma, where this is greatly expanded in the cancer. And on the right here, we have a stain. So the brown is actually showing fibroblasts. So there's this big influx of fibroblasts. They produce a lot more stroma in cancer and have a big impact. And they're known to do a lot of different things. So this is just a very brief overview to show CAFs do a lot. There's a lot of work to go into them, and they're very important cancer. But my research is more focused on normal tissue fibroblasts. So this is before they're converted by tumor cells. And this is because in early stage cancers, where we're looking into things like prevention. the tumor is actually going to be surrounded by a lot of normal tissue and normal fibroblasts. And most of this interaction is going to occur at the invasive front, where the tumor cells are sort of diving into the normal tissue, as opposed to a late stage cancer where the tumor is surrounded. And there's a bit of work showing things like the extracellular matrix of normal tissue can constrain growth. But some of my work has been showing how normal fibroblasts can actually impact tumor behavior without being cancer-associated fibroblasts. So to go back to an earlier thing, they're saying SCCs they're driven by long-term environmental damage, such as sun damage and smoking. And this is quite important in fibroblasts because these are a persistent cell type, where we have our epithelial cells have a lot of rapid growth and turnover, so these are renewed constantly. Fibroblasts don't divide as much, or they're not replicated. So these are going to be exposed quite long-term to this environmental damage before the cancer arises. And this was actually something I was interested back in my skin cancer research. So I worked in melanoma, and we all very much associate melanoma with sun exposure. But melanoma can actually develop on sun-exposed or non-sun exposed skin. And so what I was interested was, how does this affect the fibroblasts in the tumor microenvironment? So what I actually showed was the fibroblasts in the skin that are chronically exposed to UV radiation were actually profoundly different from those in sun-protected skin. And this had significant impacts on the microenvironment of the skin, which we all know sun damage does things like wrinkling and affects our tissues. But what was interesting was that when this damage had occurred and then the tumor arose in the skin, this actually altered the clinical outcomes of melanoma patients. So there was differences in survival and progression and immune cell infiltration just based on the damage that had occurred before the tumor had arisen. And if simply started looking at collagen integrity, for example, we can predict survival. So this comes back to the chronic damage to fibroblasts can alter the microenvironment before the cancer arises. And I've shown, at least in melanoma, this impacts clinical outcomes. So one interest of my preventative medicine side is, can we target tissue damage before the onset of cancer? Then this is, again, not something I've got a lot of work into yet, but something I'm definitely interested in looking in terms of smoking and alcohol damage in terms of the oral tissues. But one thing that I don't think has been getting a lot more attention lately, but in fibroblast research, not everyone realizes that not all fibroblasts are made equal. So fibroblasts from different tissues of site are quite different. And even if you take fibroblasts from the same tissue, so for example, the skin fibroblasts from your foot versus the skin fibroblasts in your leg, even these will show anatomical specific differences. And these differences usually show differences in developmental programs, so organ-specific development. And a big fact that we know is tissue-specific extracellular matrix. Just this is one thing that fibroblasts are known for. And this probably reflects fibroblasts' crucial role in epithelial cell development. So during embryogenesis and as tissues develop, as we develop, fibroblasts in the stroma actually provide positional cues to help the differentiation of the epithelial cells above them, because different epithelial cells have different specific structures. For example, our skin has a much tougher keratin layer to protect us, and our digestive tract has the villi. So basically, fibroblasts contain tissue-specific information. So therefore, different tissues are going to have different fibroblasts. And one sort of, coming from a skin background, one interest that I had with epithelial cancers was, as I mentioned before, they have a lot of overlapping features. They're all driven by environmental carcinogens. They have high tumor mutation burdens. They have similar driving mutations and similar genes. But cutaneous SCC or skin cancer has a very low mortality and a very low metastasis rate, whereas others like oral SCC and lung SCC have much higher metastasis and more poor outcomes. So the question we were asking was, what was the contribution of tissue-specific fibroblasts to these different clinical outcomes that we were seeing? So just to give you a nice visualization of this, we did some experiments. So these are some simple collagen constructs where we have a disk of collagen, and in here we have, for example, we've got dermal fibroblasts from the skin, oral fibroblasts, and lung fibroblasts. And on top of these constructs, we've put oral SCC cancer cells. And we can see that the same cell line is drastically different. Invasion into the collagen is drastically different depending on which fibroblast cell line is in the matrix. So oral and lung fibroblasts are making the tumor cells much more invasive, in keeping with their much worse clinical outcomes. And we could even see this when we put a cutaneous SCC, so normally not very invasive, not very metastatic, suddenly becomes quite invasive when we put it into matrices that contain lung and oral fibroblasts. So basically, we showed some nice studies where we found oral and lung fibroblasts relative to the skin fibroblasts is actually increasing the invasion and growth of these epithelial cancer cells. So we wondered, what was the difference between the fibroblasts based on these tissue sites? And to cut a long story short, I won't go too much detail, but we found that the fibroblasts from different tissues had different metabolic profiles. And what was actually driving the differences in these invasive patterns we're seeing were the unique lipids that the fibroblasts were producing and then secreting and giving to the cancer cells. So in keeping, we found that dermal fibroblasts, they weren't very lipid rich, they weren't producing much. Oral fibroblasts were producing what we call sphingomyelins. These were being transferred to the oral SCCs and driving aggressive cancer. And lung fibroblasts were producing a lot of triglycerides and transferring those to the lung SCCs. I won't go too much into the complexes of lipid biology, but just to highlight that sphingomyelin, which the oral fibroblasts were producing, is basically broken down through this pathway into what we call sphingosine-1 phosphate. And this is a signaling molecule that it activates all these pathways, which some people might be familiar with. But basically, these are all pro-tumorigenic pathways that can drive tumor growth, migration, invasion, angiogenesis, and things that result in more aggressive cancers. So what we were finding was these oral fibroblasts were producing sphingomyelins, which then in our head and neck SCC cells were breaking them down to activate these signaling pathways to drive growth and invasion. And just to give a bit of lung details, so the lung fibroblasts were producing triglycerides. So these are the fats that we typically think of when we think of how we store fat in our body. They're stored in lipid droplets in our cells, which you can see here in these fluorescent images. So there's a lot more of these in lung fibroblasts. They were transferring these triglycerides to the lung SCC cells, and they were using these fats as a fuel to produce cholesterol. And this was driving their invasion and growth. So we're sort of seeing tissue-specific differences in fibroblasts and lipid pathways that were contributing to similar phenotypes. And that if we looked in some clinical cohorts, these markers of fibroblasts and lipids were sort of correlating with clinical outcomes. So just looking at some TCGA data, we could see that if we looked at genes of markers of lipids or signatures to represent fibroblasts, we could see that higher expression of these markers were associated with poorer overall survival and progression, as well as looking at the genes that break down these lipids to be used. The higher the expression, also the poor outcome in lung SCCs. But as I said before, I'm more interested in the normal fibroblasts. So these were all modeled with normal fibroblasts interacting with cancer, not cancer-associated fibroblasts interacting with cancer, which means they might actually apply to earlier stage disease. So SCC development can occur through this progressive stepwise pattern of normal epithelium to dysplasia, carcinoma in situ before developing into full SCC. So this would allow time to interact with these normal fibroblasts. So we went and looked at some of these patterns in some data of SCC development. And here on the lung side of things, what was interesting, we were actually seeing the expression of these genes that are responsible for producing cholesterol, which was what they were doing with the lipids from the fibroblasts, actually progresses and gets higher through the development from normal tissue to SCC. But what was also more exciting was if we looked at patients where they had data, so this is some carcinoma in situ, so the pre-malignancy before lung SCC, they looked at those that had regressed, so didn't develop into cancer versus those that did progress to cancer. And those that progressed to cancer actually have high expression of cholesterol metabolism genes, suggesting that this lipid interaction with the fibroblasts could be a marker of patients at risk of developing full cancer. And this gives some exciting potential, especially around cholesterol, because we have a readily available and a very well-tolerated drug, statins, that most people might be familiar with. So at least in our experiments in the lab, we were able to show that these statins were able to prevent the growth and the invasion that the lung fibroblasts were inducing in the cancer, and was able to prove in survival in some in vivo models. Now, there is a bit of work around statins in cancer and some very conflicting data. But looking at lung specifically, there's one study at least that shows statin use is associated with lower risk of lung cancer. But there is a lot of conflicting results. And that might be because they're not sort of subtyping what kind of lung cancer. As I showed, this is based in lung SCC. So here's another paper that just showed statins are protective in lung SCCs, but not adenocarcinoma, another type of lung cancer. So this might be quite specific, and it could also be interesting to look at this by stage. If this is a risk factor early on, but not later on, that could impact survival and associations there. So this is something I'm quite interested in. To go back to the oral side of things quickly, not as much data on that in terms of pre-malignancy. A little bit of data showing that there is an increase in this S1P and some of these signaling pathways from oral potentially pre-malignant disease and dysplasia to SCCs. So it could be involved in earlier stages of oral cancer development as well. And there is a bit of work with one of the enzymes down the pathway correlated with outcomes and metastasis and prognosis in later stage cancers in the literature. So my data is basically suggested there's metabolic interactions that are specific to fibroblasts in specific tissues. And these could occur in pre-malignancy and early stage development. So one of the things I'm interested in is, can we repurpose approved metabolic drugs that are easily accessible, like statins, for early stage cancer prevention or prediction? So for the oral side of this thing, as I mentioned, we're seeing single-myelin signaling increasing with progression to OSCC in the data set. And in my work, I was able to show that a drug called imipramine was able to induce the invasion that was induced by this single-myelins from the oral fibroblasts. But what was interesting is imipramine is actually an antidepressant and falls into this class of drugs called phiasmas. So that stands for functional inhibitors of acid-sphingomyelinase, and actually includes 14 other antidepressants. And sphingomyelinase is actually the enzyme that is responsible for breaking down sphingomyelin to allow it to go through this cascade that then downstream signal to cancer. And interestingly, there is one study that's actually showed that out of Singapore that they found that patients taking antidepressants were actually at a reduced risk of developing oral cancer in a time and dose-dependent manner. So this piqued my interest of, well, if they're on antidepressants, and we know some antidepressants can inhibit this enzyme, are these pathways related? And there's also some evidence that this pathway is involved in head and neck development. So there's one study where they knocked out SPHK1, so one of the two enzymes that are responsible for this downstream part of the pathway. They knocked it out in a murine model of oral carcinogenesis. And interestingly, when we lose this signaling, there was actually a significant reduction in the number of mice that developed tumors, and also that they developed fewer tumors that were significantly smaller than the control mice. But there wasn't complete ablation. And this could be because they're only blocking half the pathway, whereas, as I mentioned, these drugs and antidepressants are blocking at the start. So what would happen if we were inhibiting this part of the pathway with drugs like antidepressants? Well, there is actually a link between antidepressants and sphingomyelin metabolism. So as I mentioned, the sphingomyelin is broken down through this pathway. That's a bit of a simplification. Actually, sphingomyelin is made from ceramides. So ceramide's kind of the starting process, and it can go both ways. And how these drugs work is they inhibit the breakdown of sphingomyelin to ceramide, which then builds up leads to the buildup of sphingomyelin in the cells. This stops further conversion of ceramide into sphingomyelin, which builds up ceramide in the endoplasmic reticulum, and this actually induces a process known as autophagy as a response to the buildup of ceramide. So simply, autophagy is just a process where cells can sort of engulf and eat part of themselves. So they'll get some components of cells, engulf them, what we call an autophagosome. These will be broken down and lysed into the basic building blocks like proteins and amino acids and things, which then can be recycled and used by the cell again. So this is a kind of a double-edged sword in cancer, because in healthy cells and in early stage cancer, it can actually remove damaged cell components, so it's protective and can stop the development of cancer. But later stage cancer cells are known to sort of co-opt this process, and they can break down parts of themselves for the building blocks that will then allow them to grow more and survive stress responses and things like drugs. So it's kind of an ambiguous role in cancer sometimes. But linking this back to tricyclic antidepressants, there's actually a paper similar to what I showed in oral cancer. There's also work showing that these antidepressants lower the risk of glioblastoma. But interestingly, where we don't have the data in oral cancer, there are some nice papers out of Douglas Hanahan's lab actually showing imipramine inducing autophagy in the glioblastoma. And this actually increasing survival in murine models, and it enhances the effects of other drugs they use and can even improve the immune response. So they were showing that, yeah, they're inducing autophagy. This is killing the cells, not only killing the cells, but also reprogramming the immune system to have a good immune response. So sort of to bring this together, we know antidepressants, well, at least from some studies, have showed a reduced risk of developing oral cancer. And there are other drugs out there that do this. This inhibits sphingomyelin metabolism, which I have shown drives aggressive oral SCCs when it's provided by oral fibroblasts. And this is known to induce autophagy, which at least in glioblastoma has had anti-cancer effects, inducing cell death and immune response. So what I'm interested in, will this same pathway be working in oral cancer, early stage oral cancer, and can we use some of these drugs to reduce risk? But as I said, I'm quite new to the role, so there's still a few questions to answer around this. So we still need to do some more work, sort of identify what role does sphingomyelin play in oral cancer development and early stage pre-malignancy. As I mentioned, most of my experimental work so far has been done in the more cancer cell line setting. And then can we use readily available drugs to target this pathway, or even use markers of this pathway's activity as biomarkers for the risk if a patient is going to progress or develop oral cancers? And can we do this with, as I mentioned, antidepressants, but there are a lot of other plasma drugs which are commonly prescribed drugs to work with here. But also taking back to what I said earlier, how do things like smoking and alcohol into damage to the microenvironment? Is it going to impact these metabolic pathways? Does it make them more aggressive and more worse? Is something I'm definitely very interested in. Just to wrap up and give a quick summary. So my work is very much interested in tissue-specific fibroblast lipid biomarkers of the metabolism, and sort of understanding these biological interactions to sort of understand how the cancer develops, to then identify new risk stratification methods for SAC development to utilize in patients that hopefully can predict the risk of progression or even prognosis if early stage cancer is established. And I really want to focus this around targeting metabolic pathways with repurposing approved and accessible medicines like antidepressants, statins, and things to make the medicine more accessible, to basically prevent and reduce the risk of populations at risk. Yeah, so I'd like to say thanks for the opportunity to present this work. Thanks to all my Liverpool for the opportunity to sort of set up my lab and get this work up and going here, and to all of my past collaborators who sort of helped me set up my career and get all the work that's been done I've just shown you so far, and I'll happily answer any questions.
Thank you very much, Tim. And I did put something in the chat box. Just please type in your questions or raise your hand and I can go to the questions. So I'll maybe start. I think I reviewed a PhD in 2021 or 22 looking at Mendelian randomization as a way of exploring cholesterol lowering as a way of preventing oral cancer. So there's this
Yeah.
epidemiological association. They used Mendelian randomization. They looked at data from GWAS studies, I think on a couple of 100,000 patients. And what they come up with was really confusing in the light of your work, because they say, yes, it looks real, but it's independent of the cholesterol lowering effect. And maybe that's just the limit of Mendelian randomization as a technique. But are you familiar with that?
Not with the technique, but there are a few things to consider. I think I've thought about this for a while with statins as well, in terms of the because I mean statins' main target is the cholesterol that your sort of liver processes and then the apolipoproteins. Whereas this is going to be a localized, the tissue fibroblasts are giving the fuel for the cancer cells. So unless we can get the statin drugs affecting the cancer cells in the localized tumor, this might not be seeing an effect, because I think 90%, well, I guess it depends on the statin you use, but some of them, I think 90% of it's metabolized in the liver. So it could be an association between circulating cholesterol levels and the cholesterol levels that are just occurring in that localized tissue environment. But yeah, I'm not a big expert on population science and things like that, so I don't know exactly how these control studies work exactly and what limits they've got. But
they're looking at, I think, at patients' inherent way they deal with so the genetic variants that alter lipid and cholesterol metabolism, and looking at that, they can see whether the effect is reproduced in patients that you would expect to have lower lipids, lower cholesterol from their genotype. And looking across big studies, they're held to be quite powerful. So the Bristol group and George Davis Smith was kind of like a pioneer of this as a way of looking at lots of things. But as you know, we run an oral chemo prevention trial from Liverpool, which is a national trial looking at a repurposed drug that's been used for other things. And in the first instance, sodium valproate. And we've got potentially a pathway that we can plug other candidates in this area of repurposed drugs off license is quite trendy in cancer prevention, isn't it?
Yeah. I mean, I think I believe there's some work showing the valproic acid also inducing autophagy in settings. So it could be interesting if there's an overlapping mechanism there between
well,
the
that
two.
trial ends, where are we? 19th of November. Yeah, we've got 5 days to recruit patients and then it's closing.
Yeah.
So that's been a long haul, but we got the study done. So other questions for Tim? I can invite, Keith's got a question. Keith Hunter, we know that fibroblast phenotypes change rapidly when grown in culture. Are these metabolic changes seen in vivo? Can you identify it in single cell data?
Yeah, so a lot of my work's been done in just tissue culture settings, which, I mean, we all know that doesn't necessarily mimic what happens in the body. I've done some work with looking at these in patient samples, spatial transcriptomics to look at stromal epithelial interactions, and we see similar things there. I'm very much interested going forward in my research, looking at, how, if we can get these tissues, fibroblasts from fresh tissue, how they do adapt and pathways will change, adaptation to culture. I've often had trouble looking at metabolic pathways in single cell data. A lot of the genes just aren't picked up, and I'm not sure if that's just because they're not expressed at high levels. With single cell, there's the problem of looking at when you look at metabolism, the gene expression doesn't always equal the production of metabolites, because it's a flux, not the more gene doesn't necessarily mean more metabolite. So we can sort of kind of infer what's going on based on the expression of the genes, but knowing how that's going to directly correlate to the metabolites they're producing and how that's then affected when we dissociate the tissue, it's kind of something quite hard. So we do sort of need to rely on these in vitro and vivo models. I've done some in vivo work, and we do see similar phenotypes with the different fibroblast cell lines in the tumor cells in vivo, and they respond well to things like statin as well, so lowering the cholesterol.
Yeah, and it's interesting how suddenly this story of lipids come from two different directions. I was having a long conversation with Peter Waitman from the physics group, and he'd been working with Sarah Copeland's group on uveal melanoma. And you may know from Peter's group, it basically looking at the chemical profile of cells and how that might correlate with phenotype or behavior or metastasis. So the question was about liver metastasis, yes or no, from uveal melanoma. And the number one chemical entity that he discovered from his very agnostic approach, looking across all potential molecules, was, guess what? Lipid.
Yeah.
And then Sarah Copeland said, that's great, because we found that lipids important in these tumors separately by a more conventional molecular biology approach. So it's really nice when your top hit comes and matches one of our top hits. And then Tim gives us a lecture on 4 different cancer types and talks about lipids as well. And so it all fits together well. And I suppose the thing about, whether the antidepressants or whether the cholesterol drugs, HMD CoA reductase and so forth, the advantage there is if you've got good in vitro data, because these drugs are safe, you can actually jump from really solid in vitro data from organotypic models straight into the clinic.
Yeah.
Because the animal work probably won't add so much, because the models are flawed in animal work. And actually, if the drugs are safe, these can be explored in window of opportunity studies like the SAVOR trial. So we've actually got a quicker way to clinic because of the safety profile, provided the epidemiological evidence is there and we can back it up with some cell biology as well. I think that's quite intriguing for our group in particular, because we've sort of accidentally pioneered something with sodium valproate and managed to get it into clinic. Now, whether it works or not, another issue, but we've created a model and a process. So Andrew Shaki's asked a question. You pointed out the impact and importance of carcinogens on fibroblasts in the tumor microenvironment for tobacco and smoking. Would that imply that comparable fibroblasts in HPV positive cancer might be more normal? So the difference in prognosis between HPV mediated and non-HPV mediated head and neck cancer, could it all be about the fibroblasts?
Yeah, I mean, that's something quite interesting. I think it's a good setting I could use going forward, because I mean, with melanoma, I was lucky there was, you've got sun-damaged skin and non-sun-damaged skin to compare. But if a lot of these cancers are induced by tobacco smoking, then using the HPV positive where you wouldn't have that, and comparing the fibroblasts between the two would be quite interesting to see, sort of decouple that contribution of the damage. In terms of, I guess, my model and what I've shown, the specific lipids, we've used normal fibroblasts from a combination of some donors and some commercial ones, but never really had the smoking status or alcohol consumption status for them. So we don't really know what the contribution there has been. So most of my sort of work has been, in terms of damage, has been on the UV side of things. And I would sort of like to see that does translate in terms of smoking and tobacco and alcohol. And I guess, yeah, if fibroblasts are doing different things or the same thing within HPV cancer, positive and negative, would be quite interesting.
Yeah, for sure. So we've got quite a lot of tissue and matched pairs and cell lines. Keith just made that point. We've probably got a lot of tissue that you can, and a lot of cell lines and models that you could interrogate. And I think, Tim, we look forward to having seen that data. Really intriguing of you interrogating our head and neck cancer models in the lab with your hypotheses and your drugs of interest and your mechanism of interest. That's great. Francis has asked a question, Francis Greeny. She's just maybe a comment. We've got primary derived fibroblasts available from both oral cancer, HPV positive and negative, if you want them. So myself, Janet Risk, Francis was involved, myself and Janet Risk supervised a PhD student, 10 or 12 years ago, and we did a lot of primary cell culture, and a lot of those fibroblasts grew very well, and the epithelial component of the tumors often grew a little bit less well unless the tumors were very aggressive. But the fibroblasts, I think, are frozen down. I think we've got quite a lot. And a lot of them were from standard oral cancers. So there we go. And Keith, Jetsy's got them all outgrowing. So there we go.
I'm in a very good place to do this work. It's a lot of resources. And
And I think we could get lots more, because I think the PhD student who did it was relatively new to it and managed to get it going fairly well from a fairly simple explant model. So we got a cell culture lab temporarily, and the facilities out at Aintree, and we managed to bring that work into the lab. So if you want to crank that handle from patients through to your lab, we've got the permissions, we've got the processes in the past. Certainly, they should be able to reinvigorate them. Yeah. That's superb, Tim. We've taken our 45 minutes, so that's great and really interesting. Look forward to seeing your results in the future, and glad that you're able to join us in Liverpool. Just as a reminder for everybody here, next week's lecture, back to the boring clinicians, but it's not at all boring. We've got Vin Polleri talking from the Institute of Cancer Research and the Royal Marsden, so I'm sure you'll all want to join us. It's the same time, same place, 7 o'clock Tuesday night on Zoom. Thanks very much, everybody. Cheers.